Hi, my name is Karolina. I'm a virology PhD student, and because I believe virology is full of fascinating stories, I decided to share some of them. Today we know that the AIDS epidemic originated in Africa, already in the early 1900s. But for a long time, the disease stayed under the radar, and it only became noticed for the first time in the 1980s in the United States, when groups of previously healthy people began to suffer from rare diseases that their immune system was unable to fight off. These people were during their lives infected with the HIV virus, and later they developed a disease called AIDS, meaning that their immune system basically stopped functioning properly. That's why the very first reports didn't speak about noticing some new virus or describing the viral particle. Instead, they talked about unusually high numbers of previously healthy people suffering from rare diseases. The very first case report came from the CDC, the American Center for Disease Control, and it talked about just 5 patients suffering from a rare case of pneumonia caused by a fungi called Pneumocystis. And while the fungi is very common and it is present in the environment, developing the disease is very rare, and typically it only happens in immunocompromised patients. These detected pneumonia cases were just 5 cases that happened across several hospitals in Los Angeles, but they were noticed and connected thanks to an observant technician working for the CDC. And that's because of a drug that was used to treat these cases. The medicine used for this pneumonia, pentamidine, was initially developed to treat sleeping sickness, and it was listed as an experimental drug. Because of that, it wasn't something available in the pharmacies, and it could be requested only from the CDC. These requests were typically very infrequent, but suddenly there were 5 of them. These 5 patients had several things in common. They were all previously healthy, they were around 30 years old, they suffered from unexplained immune deficiency, and they were homosexuals. There were no connections between them, and they didn't know each other. And by the time the report came out, 2 of the 5 patients had already died. This first report of strange medical cases was soon followed by another one, just 2 pages long report, describing even more cases of this rare pneumonia. And this time, these new pneumonia cases were joined by another suspicious observation. This time was discussed also unusual number of cases of Kaposi sarcoma. Kaposi sarcoma is a type of cancer that typically appears as tumors on the skin. It is associated with infection with another human virus, with herpes virus 8. But being infected with this herpes virus doesn't necessarily mean that the person will develop this cancer. Actually, it is quite common infection. In different areas of the world, between 10 but up to 50% of people are infected with this herpes virus. But the number of cancer cases is very low, because once again, the risk is associated with issues with the immune system. It is a very similar situation to the pneumonia cases caused by the Pneumocystis fungi. In people with healthy immune system, herpes virus 8 is kept in check by the immune surveillance. But when the immune system is compromised, which in these cases was caused by AIDS, it allows the virus to reactivate and proliferate, which leads to the development of Kaposi sarcoma. So this second CDC report described an unusual number of Kaposi sarcoma cases in the population of previously healthy gay men who had immune deficiency and who were also much younger than typical Kaposi sarcoma patients. Interestingly, These earliest reports consisted of two parts. The first one was a simple description of the cases. But the second part was an editorial note, speculating about possible causes of these diseases or their appearance in these populations of young patients who were previously healthy. In these editorial notes were pointed out several key facts. Firstly, since these cases were described in the population of homosexual men, there were suggestions about the possible sexual transfer of an infectious agent in certain communities. And as we know now, HIV is sexually transmitted virus, so this original assumption was correct. And secondly, these editorial notes discuss the connection of the immune dysfunction observed in these patients with the detected rare infections. And they speculated about what could have possibly caused this immune deficiency. And these earliest reports already suggested that maybe the immune system of these patients was negatively affected by some kind of infection. Of course, the HIV virus was unknown at the time, and they were speculating about the possible role of human cytomegalovirus. So they were wrong about the specific agent, the specific virus involved, but they still got very close to the truth. So, these were the first noticed AIDS patients, but soon more detected cases followed. By the end of 1981, about 300 cases of severe immune deficiency had been reported, and these were mostly among gay men. And by the end of the year, almost half of these patients died. And in the years to come, both of these numbers continue to grow. So the first task in facing the starting epidemics was to determine how the disease is transmitted. This information could also provide some insight into the cause of the disease. Today we know, since we have the advantage of living in the future, that AIDS is caused by the HIV virus. But some of the early theories considered that it could be triggered, for example, by drug use or by some lifestyle factors. In the 1980s, as more and more cases were identified, researchers began to notice patterns in AIDS transmission. As I mentioned, already the earliest reports of AIDS speculated about the role of sexual transfer. Originally, the transmission was detected mostly among gay men, but later it became clear that AIDS is transmitted also among heterosexual couples. I actually remember that a few years back I read a memoir by a researcher who worked in Africa, and at one point he was trying to publish his study about the transmission of AIDS in heterosexual couples. But his research paper got rejected by the editor with the claim that everybody knows HIV is a gay disease. Just imagine the frustration. But coming back to the early observations. In addition to gay men, other groups soon began to show similar immune deficiency. One of these groups were intravenous drug users, which provided another important hint that the disease is transmitted through blood. Other affected group were hemophiliacs, a group of people for whom it was necessary to receive regular infusions of clotting factor concentrates prepared from donated blood. But before the HIV virus was identified and before blood screening tests were available, many blood donations were unknowingly contaminated with HIV. These clotting factors concentrates were often made by pooling plasma from thousands of donors. So if even one donor was HIV positive, the entire batch of clotting factor could become contaminated. As a result, approximately 15,000 hemophiliacs in the US became infected with HIV between 1981 and 1984 as a result of transfusions with contaminated blood products. But the blood products used to treat hemophiliacs were filtered to prevent bacterial and fungal diseases. So, after defiltration, bacteria and fungi should be cleared out. However, and this is a flashback to episode 2, the filters weren't small enough to protect also from viruses. So the fact that hemophiliacs could be infected by their treatment suggested that the disease has a viral origin. While the researchers were growing their understanding how AIDS is transmitted, they were also searching for its causative agent. And a crucial moment in the understanding of AIDS came with the discovery of the HIV virus. The virus was isolated for the first time by researchers from a French group led by Luc Montagnier. In 1983, these scientists isolated and cultured cells from lymph nodes coming from a patient who suffered from a condition typical for an early stage of AIDS. The scientists maintained these isolated lymph node cells in cell culture, and after 15 days, they could detect reverse transcriptase activity in the culture medium. Now, reverse transcriptase is not naturally found in human cells. The presence of reverse transcriptase activity is instead a hallmark of retroviruses, also distinguishing them from other types of viruses. So it was a sign that the isolated cells are infected with a retrovirus. The French researchers were able to isolate the virus and show that it infected CD4 positive T lymphocytes, so the very cells affected in AIDS. They also used electron microscopy to visualize the viral particles budding from the infected cells, and they show that this virus is distinct from the two already known human retroviruses. Luc Montagnier and François Barré-Sinoussi were later, for this discovery of HIV virus, awarded the Nobel Prize. Their study was soon followed by another report from an American lab led by Robert Gallo. Gallo's team was also able to isolate HIV virus from patient samples, and the isolated virus was consistently found in individuals with AIDS or in those who were at high risk of developing AIDS, so hemophiliacs, gay men, and intravenous drug users. This epidemiological correlation suggested there may be a causal link between the virus and the disease. Gallo's team also developed important tools to study HIV. For example, they prepared cell lines that could grow the virus. And most importantly, they developed a blood test that could detect antibodies to the virus. So a test which could screen for HIV infection, and this test also allowed the screening of blood supplies for HIV. So it prevented further transmissions of AIDS by blood transfusions. The discovery that HIV virus is the cause of AIDS brought up another question. Where did the virus came from? Interestingly, the early days of HIV pandemics gave us a phrase that everybody who watches movies knows today: patient zero, aka the very first patient starting the spread of a disease. The term patient zero became widely known in the context of HIV epidemics, but it was used wrongly, it was scandalized, and it caused a lot of harm. The label Patient Zero was given to a Canadian flight attendant, Gaëtan Dugas, who died of AIDS in 1984 when he was 31 years old, and he was blamed for the spread of HIV in North America. This whole idea began with a misinterpretation of a study from the early 1980s. In this study, CDC was tracing sexual contacts between gay men with AIDS to understand how the disease was being transmitted. So basically, they were drawing a network of sexual contacts among patients. I believe that the potential of this study was somewhat limited from the very beginning, because at the time it wasn't known the incubation period between HIV transmission and the development of AIDS. Today we know that it can take 10 years or even longer for AIDS to develop after HIV infection. But at the beginning of the pandemics, it was believed that the progression from exposure to AIDS happens much faster. So what it means is that if you study sexual contacts that happened, for example, a year before these people developed AIDS, you are not studying the transmission of the disease, but instead you are looking at people who were already infected for many years. And furthermore, the term patient zero was actually a misinterpretation. In the CDC study, Guyton Dugas was labeled as patient O, with the letter O standing for out of California, because he did not live in the state. But the letter O was misread as a zero, turning patient O into patient zero. This misreading of the label led to the myth that Dugas was the original source of HIV outbreak in the United States. The story of Guyton Dugas as so-called patient zero was popularized in a book written by Randy Shields, who basically painted Dugas as a villain who refused to stop having sex despite being HIV positive, and even as someone who was spreading the disease intentionally. The story was picked up by many journalists, and it led to headlines such as "The Man Who Gave Us AIDS," using Dugas's name and printing his picture, or nicknaming Dugas the Columbus of AIDS. But many people who knew Dugas during his life claimed that his presentation in Shields' book was unfair to him, and that it misrepresented his real personality and his real behavior. And as for the claim that Guyton Dugas was the person who brought HIV and AIDS into the US, this suggestion was debunked by later research. A group of scientists studied the oldest HIV sequences from the United States obtained from archived blood samples, already from the 1970s, which turned out to be HIV positive. And they also got a sample from the very HIV virus that infected directly Guyton Dugas. They compared the sequences between each other and also with other known HIV sequences. And their results showed that HIV spread to the US from the Caribbean, and that the virus that infected Dugas was a completely typical US strain at the time. Nothing suggested that Dugas played a unique role in the spread of the virus, and they found no evidence that the so-called patient zero was the first person infected with this type of HIV. So what can study of HIV sequences tell us about the real patient zero and about the origin of HIV virus? It turns out quite a lot, as long as you manage to collect appropriate samples. One question that can be answered by sequence analysis is the age of a virus. Because as viruses replicate, their genetic sequence collects mutations, and scientists can use this. It is possible to look at viral sequences and use a method called molecular clock to estimate how old the virus is. The basic idea is that mutations happen at a relatively steady rate, much like a clock ticking at a consistent pace. This means that by counting the number of differences between the virus's genetic sequences, you can estimate how long it has been since the virus started spreading and started mutating. To estimate the beginning of the HIV pandemic as accurately as possible, it was important to locate and sequence some old historical HIV samples. A crucial role played the discovery of HIV-positive plasma sample that was collected already in the year 1959 from an HIV-positive man in Kinshasa, a town in Congo. Because of the age of the sample, it was very tricky to isolate the viral genetic information, because over time, the viral RNA degraded and there wasn't much left. But the researchers were able to amplify and sequence at least just 4 short fragments of the viral nucleic acid. And a few years later was discovered another historical HIV sample. Researchers were studying paraffin tissue blocks that were prepared and stored for years, also in Kinshasa, and they screened them by PCR for the presence of HIV RNA. And indeed, they discovered HIV RNA in a lymph node biopsy collected from a woman in 1960. And they were able to isolate and sequence 12 short fragments of the viral genetic information. These 2 HIV samples, one from the year 1959 and the second one from 1960, represent the oldest known HIV sequences in the world. And it turned out that they are surprisingly different. They differ by approximately 12%. Now, this is a considerable genetic difference. And what it suggests is that HIV was evolving in humans for decades before these samples were collected. Comparison of these sequences place their most recent common ancestor near the beginning of the 20th century, probably sometime after the year 1908. So this showed that the diversification of HIV virus in Africa happened decades before AIDS was even recognized. So these studies of sequences gave us some idea when did the virus infect humans for the first time. And it was again the study of viral genetic information that revealed how this first transmission likely happened. Scientists noticed that monkeys are infected with a virus very similar to HIV. While the name HIV means human immunodeficiency virus, the monkey version was named SIV, standing for simian immunodeficiency virus. The similarity between these viruses suggested that the human and monkey versions are closely related. There are more than 40 different species of African monkeys that are infected with SIV. But each of these species has their own species-specific SIV virus. Now, some of these SIV viruses are more similar to human HIV, others less. And the virus that was the closest and most similar to HIV was discovered in 1989 in a chimpanzee. This, of course, led to the idea that HIV is of chimpanzee origin. But it wasn't so easy to prove it. Because for a long time, this virus, very similar to HIV, was only found in chimpanzees held in captivity. So what followed for the next 10 years was testing of more and more chimpanzees and searching for further examples of this SIV virus. But after screening 1,000 more chimpanzees, scientists only found 1 more infected. So it appeared that SIV is extremely rare in chimpanzees. And it seemed possible that maybe they are not the source of the disease, but instead both people and chimpanzees were infected from some other source, presumably some other African monkey. But later it turned out that the issue lied somewhere else. I would say that the issue was that the researchers had a really bad luck. Because even though more than 1,000 chimpanzees were tested, most of them belonged to 1 subspecies. And, of course, it was the subspecies which doesn't get infected with SIV. The issue could finally be resolved after development of new techniques that allowed non-invasive screening for SIV. Because originally, all SIV viruses or the virus isolates, were obtained from blood or tissue samples. Now, this of course limited the extent to which rare and endangered species could be tested. But new techniques allowed to detect SIV in urine or fecal samples. This meant more monkeys could be tested, and even the ones from endangered species. And in one particular chimpanzee subspecies, Pan troglodytes, was detected the SIV infection so highly similar to HIV. It is believed that this was the virus that gave the origin to the human HIV pandemics. The SIV virus was probably transmitted to humans through exposure to infected blood. So, probably through activities such as hunting or during the butchery of bushmeat. The initial transmission probably happened in some injured hunter or cook whose blood got in contact with the blood of infected primate. Based on the study of the viral sequences, this happened probably at the beginning of the 20th century in Cameroon. From this original transmission, the virus started to spread, and for a long time the infection managed to stay under the radar. Probably because many other infections were affecting people in the area and there was limited access to the health care. And the study of HIV genetic sequence revealed even more interesting information. Most importantly, that the transmission of HIV from primates to people didn't happen just once, but instead it occurred several times. So instead of imagining just one bleeding hunter who injured himself when processing bushmeat, you should picture at least 12 bleeding hunters. These different transmissions generated several HIV types. But not all of these different groups of the virus contributed to the pandemic in the same way. There are two main HIV lineages, HIV-1 and HIV-2. HIV-1 is the most important type, especially its group M. This is the virus that got transmitted to a person from a chimpanzee. The virus that was detected in the first AIDS descriptions. The same virus that infects millions of people, and it caused more than 90% of all HIV infections. But the lineage HIV-1 contains also other groups. Group N started as independent transmission of HIV-1 from a chimpanzee, similarly to the pandemic group M, but it remained confined in Cameroon, and overall were detected only about 20 cases of group N infections. Next, there are group O and P. Genetic studies indicate that these viruses did not originate from a chimpanzee, but instead they are most similar to the SIV variant infecting lowland gorillas. So we also had HIV transmissions from gorillas. Group O is the more spread of the two, infecting more than 100,000 people. On the other hand, Group P cases are extremely rare. Then there is another HIV lineage entirely, the HIV-2 virus. This virus was discovered in 1986, and it was different enough from HIV-1 to be classified as a separate type of HIV. And while it seemed distant from HIV-1, it was closely related to SIV virus infecting Sooty manga bay monkeys. Now, I had no idea what kind of monkeys they are, so I looked them up, and they are the small and adorable kind. There is a cute YouTube video of Sooty mangabees pranking gorillas by jumping on their heads, which led me to Google if they are endangered species, and yes, they are endangered, yeah. Sooty mangabees are also frequently hunted as agricultural pests in West Africa, so there were probably many opportunities for transmission. And just like with HIV-1, the transmission of HIV-2 also happened several times. but only 2 groups of the virus managed to spread and infect more people, and the other groups were found only in a single person. Overall, HIV-2 played less important role in the pandemic than HIV-1. Both of the infections progress to AIDS, but HIV-2 progresses slower. It is also less transmissible than HIV-1. For example, there is almost complete absence of transmission from mother to child. Overall, the prevalence of HIV-2 is decreasing, and the infection is being replaced by the HIV-1 lineages of the virus. So these discoveries elucidated the past and the origin of the virus. But there was a more burning issue to solve. Initially, there were no treatments available for AIDS, the disease was spreading, and people were dying. The progression of HIV infection to AIDS has several stages. First one is the acute infection. This phase happens soon after infection, and it lasts between 2 and 4 weeks. After the initial HIV infection, people experience only mild flu-like symptoms. But during this stage, there is also a high level of viral replication, and the virus is destroying immune CD4 T cells. However, at this point, the immune system eventually mounts a response against the infection and manages to reduce the viral levels, which marks the entry into the next phase, which is the latent infection. During this time, the disease slowly progresses, but somewhat under the radar. HIV continues to replicate at lower levels, and people may not exhibit any symptoms. This stage lasts for several years, but during this whole time, the virus is actively destroying the CD4 T cells. So throughout the years, the immune system is systematically weakened. After approximately 10 years of the latency, the disease progresses into the final stage, AIDS. This happens when the number of CD4 T cells falls below a critical level, and the immune system becomes severely compromised. With the immune system not functioning properly, people become susceptible to opportunistic infections, which were the primary cause of death among the AIDS patients. During the early years of AIDS pandemic, treatment options for people with AIDS were extremely limited, only to drugs targeting these opportunistic infections. But to develop efficient therapies for AIDS, it was crucial to study the HIV replication, because understanding the process allows the development of antiviral therapies specifically targeting the virus. The HIV replication cycle begins when the virus uses its envelope proteins to bind to the CD4 receptor, allowing the virus to fuse with the cell membrane and enter the cell. Once inside, HIV releases its RNA genome and uses an enzyme called reverse transcriptase to convert its RNA into DNA. This viral DNA then integrates into the host genome with help of another viral enzyme, the integrase. The host cell machinery is basically hijacked to produce new viral RNA and proteins. And these new viral components are assembled into immature virus particles that bud off from the cell. The final step involves the enzyme protease, which processes the new virus particles into mature, infectious HIV, ready to infect another cell. Importantly, each step of this viral infectious cycle presents a target for potential therapy. For example, drugs could be targeted against the reverse transcriptase, which is necessary to form viral RNA into the DNA, or against the integrase, without which the virus cannot integrate its genome into the host DNA. Or you could target the protease that is necessary to process the immature HIV particles. The first drug against HIV was approved in 1987. It is called AZT, and it was originally developed as a drug against cancer. But if I understand it correctly, it wasn't very efficient against cancer. But it was found that AZT can inhibit HIV replication by targeting the viral reverse transcriptase. Because AZT is an analog of thymidine, so it basically mimics one of the nucleotides that make up DNA. During the reverse transcription process, the viral reverse transcriptase incorporates AZT into the DNA strand instead of thymine, which blocks the addition of further nucleotides. So AZT basically stops the synthesis of viral DNA. The treatment could substantially decrease the viral load in patients and, for example, lower the chance of HIV transmission from mother to child. But these benefits were only short-lived. In almost every patient, the amount of virus returned to the previous level within just few months, because the virus mutated and it gained resistance to the treatment. Another issue was that the drug had severe side effects, including nausea, anemia, muscle damage, and much more. So the treatment itself was challenging and painful, in addition to not being effective in the long run. I saw a picture of badges people were wearing with the inscription, "I survived AZT." And it was one of the symbols used by the community affected by HIV and by activists in their fight for development of better treatments. From the treatment point of view, the use of AZT brought us one very important lesson: that treating HIV with just one drug is not enough, because the virus quickly mutates and gains resistance. So in the following years, two very important things had to happen. The first one was development of more drugs and different classes of drugs. And the second important thing was to use these drugs in a combination therapy, where the patient is taking multiple different drugs at the same time, because this combination therapy decreases the chance that the virus will mutate and manage to escape the treatment. First, were developed more nucleoside analogs, again targeting the viral reverse transcriptase. The use of these drugs in combination had much better results than a single drug therapy. But even with the use of two drugs, there were again the issues with the virus developing resistance. Then in the following years were developed new therapies targeting different aspects of the viral replication. And it was 3 drug combinations that truly turned the tide around. For example, the combination of a protease inhibitor with 2 reverse transcriptase inhibitors. The triple combination was a life-saving therapy that completely changed AIDS. It turned it from a lethal disease into a manageable, lifelong chronic infection. But the efforts did not end there. Another mark of progress was reducing the amount of pills HIV-positive people have to take. The triple drug therapy was efficient, but it wasn't easy to follow. For example, the used prodrugs were not very stable in the body, and they were quickly metabolized. So it was necessary to take the pills every 6 to 8 hours. In 1996, a patient would have to take more than 20 pills every single day. When we had the open house at our institute, we were showing people some of these older HIV drugs, and they were also really huge pills. You would not want to take 20 of them in a day, let alone every single day for the rest of your life. Eventually, the treatment was made more simple by developing more efficient drugs, and also by combining multiple drugs in a single pill, so that the patient has to take just 1 pill a day. And these new drugs also don't have the terrible side effects associated with the early therapies. And the efforts to improve the treatment still continue. And currently, it is desired to achieve even less frequent dosing. The idea is that instead of taking a pill every day, the patient could receive an injection of a long-acting antiviral drug every few months. Promising results provide tests of capsid inhibitors. The viral capsid plays an important role in many different steps of the viral infectious cycle. So drugs targeting capsid get several opportunities to inhibit the viral replication. Currently, the newly developed highly potent capsid inhibitor can be administered as an injection once in 6 months. But it still cannot be used on its own, because again the virus would develop resistance. So to fully use its advantage, it is first necessary to develop also other drugs that could be administered only once in several months, so that these drugs can be used together in combination therapy and administered with this low frequency. Okay, but what about a cure? Thanks to the available treatments, HIV-positive people can lead a long, normal life. But the therapies I talked about don't lead to a complete cure, and if the person stops taking the drugs, the viral load rebounds back to its previous level. This happens because HIV can basically hide in a dormant state. Shortly after the infection, HIV establishes a latent reservoir by integrating itself in a subpopulation of T lymphocytes with long longevity. And because the antiviral drugs are targeting actively replicating virus, they cannot eliminate it from these inactive latent reservoirs. So even when the therapy reduces the viral load to undetectable levels, the latent virus can persist and reactivate if treatment is stopped. Currently, researchers are trying to develop strategies to fight these issues. For example, by so-called kick and kill approach, where the first step is to kick the virus, to reactivate the dormant HIV latent reservoirs, making it visible and vulnerable to the immune system and to therapeutic agents, so that it can be eliminated in the kill step. So kick and kill is a great idea with a great name, but despite some clinical studies ongoing, at this time, a true working cure for HIV does not exist. Besides the development of cure for HIV infection, another great challenge is development of a vaccine. HIV has a high mutation rate, and it is quickly changing its surface proteins, which makes it difficult for the immune system and thus a potential vaccine to target it efficiently. Additionally, the development of universal vaccine is also complicated by the diversity of HIV subtypes and strains. So despite decades of research and numerous clinical trials, so far, no working vaccine against HIV exists. But while there is no vaccine to prevent HIV infection, there is another way, because the antiviral drugs can also work for prevention. When HIV-positive person uses the antiviral therapy, their viral load is reduced to undetectable levels and they are not infectious. Besides that, the therapy also prevents the transmission from mother to child. And there is a second way how the therapy can prevent transmission. Because even HIV-negative people can take antiviral therapy as a pre-exposure prophylaxis to prevent possible infection. So basically, they can use the same therapy as HIV-positive people, which was also made possible by developing drugs without the dangerous side effects. And, like 2 weeks ago, there was a study published using the long-acting capsid inhibitor I mentioned earlier. This study was performed in South Africa and in Uganda, and for 2 years, a group of HIV-negative women received the injection of this therapy every 6 months. And they got great results. During the 2-year period, not a single participant got infected with HIV. And that is the story of HIV. Since its first identification, nearly 3 decades ago, HIV has had a profound global impact, infecting at least 60 million people and resulting in over 25 million deaths. Today, I would say that a good portion of the major challenges has been solved, but we are still far from living in an HIV-free world. One of the primary challenges today is treatment access. Specifically, detecting HIV-positive individuals and ensuring they receive the necessary treatment. Currently, around 40 million people are living with HIV worldwide, and of these, 90% know they are infected, and 77% have access to antiretroviral therapy. So basically, the other way to put it is that a quarter of HIV-positive people are not receiving treatment, which is a lot. The problem is most pronounced in Africa, which accounts for 2/3 of global HIV cases. Besides the treatment access, the other challenges of course include the development of a vaccine and of a complete cure for HIV. And to stress out that we are not out of the woods yet. And, to end the episode on a depressing note, in 2023, more than 500,000 people in the world still died as a result of complications associated with AIDS. And for now, that is all about HIV. As always, thank you so much for listening all the way through. It makes me very happy. Also, big thanks to everyone who rated or recommended the podcast. I really appreciate it. The sources for the episode will be listed in the episode description. And for the next episode, I think I will stick with HIV, but focus specifically on the story of the Berlin patient that I omitted today. If you want to be notified, you can follow the podcast in your podcast app of choice or also on Instagram. And once again, thank you for listening.